Okay, so when I put this together, I might have underestimated or overestimated how much I had to reduce in order for you to have a practical. All right, but the the basic premise, no, wrong. I'm sharing the wrong one. Okay, just give me a second. So the basic, what do you call it? Let me just escape. So for module 6, the reason why I felt that module 6 was important is because you have to understand scale, right? Mm, okay. You need to understand scale. You need to understand scale. And I'm sure many of you are asking the question, when do I use 2 micron, 8 micron, and 16 micron? There's always this thing everywhere you go. The first thing is you hear it's a 2 micron resolution. But if you notice, all the data sets that are being shown is at 16 micron, right? So what is scale? When do you use scale? All right. What is bin size? When do you use bin size? So bin size is very, very different from scale. They are used very interchangeably. All right. And it's okay, all right, to use it interchangeably as long as you understand, all right, what is the potential of your bin and your scale. So that is the take-home message, because that is going to actually allow you to perform and what do you call it? To make smart choices about segmentation, to make smart choices about a platform. All right. There was someone who came just last week and a few weeks ago, and sometimes I just tell them, why do you want to invest in a Visium HD when you can simply just get a similar result or even better results from a Visium? All right. So these are all things that you already have all the skills of a scientist. Like, we don't have to teach you any of this. All right. But what we want, all right, for you to take home from these lectures is to understand the value of these spatial platforms and to make all the right choices. All right. And it's not easy, right? You can just, I mean, basically there are tons of tutorials out there. You can read and you can try them out. But it's nothing like having a group of people together and discussing and sharing ideas and sharing problems, especially. So that's how we solve things. All right. Okay. So why is scale important? So on the left, this was the image that I took recently, about 2, 3 years ago, as a postdoc here. And on the right is the image that I took when I was a graduate student at the Neuro and in Montreal. So this is where it all started, at least for me. All right. And this is where everything starts, even for you guys, right? Why do you want to do spatial? Because you have seen, you have observed something from an image. You have observed something, maybe even from single cell, even if it's not an image, you have observed some kind of a relationship, right? But the thing with single cell is the relationships are all based on gene expression, which means that you can have similar cells which have the same, which are close together, all right, when it comes to gene expression distances, but they might not be close together in an actual tissue context, which means that their function would be different, right? So that is why you want to understand. And another thing why scale is important is it also depends on what kind of organization you want to understand. So remember, when we went through Banksy, some of you were asking, why do we need then to do spatial? Why do we need to fine tune the parameters? All right. Why is parameter fine tuning important? Why is domain structure so important? Why is it important to understand whether there is a need for domain identification? It really depends on your tissue context. So here on the left, all right, you have a single blood vessel. But you look at these, the way the tissue or the way the cells are structured here, as compared to the packed nature of your cells at the bottom right, at the bottom left. When you look at these images, how many of you actually ask yourself these questions? So in one of I think it was Star Map. So if you read this paper that I read, I can't remember. I can't seem to recall the author. So the thing is, when you look at tissue in 2 dimensions, you find that what they found is that excitatory neurons were actually neighbors with their own kind, which means with other excitatory neurons. But when you reverse the Z, all right, excitatory neurons are actually no longer in close neighborhood with their own cell types. They're actually their neighboring cells become inhibitory cells. So these are things, all right, these are the things that you want to understand and use spatial for. But the thing is, scale becomes important because it will determine the distance or the resolution. If your scale is small, 2 micron, yes, for sure, you can understand what is subcellular. You can identify your single cells. But we change the scale when it comes to Banksy. So in Banksy or in any kind of spatial clustering, all right, the scale is no longer the bin size or the resolution of the technology that you're using is based on the question that you want to ask biologically. So if you want to understand, all right, and you want to easily, for instance, identify the layers in your human brain or in your normal mouse brain, then your scale becomes larger. All right. So understand what your scale is. So scale can be driven by the platform. So in this case, we are all learning about Visium. We're using Visium HD as a platform. So it can be 2 micron, 8 micron, or 16 micron. But that scale, what does it mean when it comes to applying it over, all right, your query? What is the value that you get? That scale is a different scale. So you need to know the difference between these before you actually jump in and say, I want a 2 micron or an 8 micron or a 16 micron. And also, we're not doing this here, but Cell Ranger, you can do this yourself actually. You can even write a simple code and do it yourself. You can alter the bins. It doesn't have to be 2, 8, and 16. It can be 2, 4, 12, or 24, any multiples, as long as it has to be, it can even be, I'm not sure if it can be a rectangle, but yes, you can change the size of your bins. So evaluate all of this, and all of this, all right, should be guided by how much you understand your tissue structure, all right, and the layout of your tissue. If, all right, your main objective is to study blood vessels, maybe your bin size should actually be guided by the average size of your blood vessels. I mean, who says that you can't do that, right? There's no standard rules when you want to understand or when you want to do any kind of science, right? So these are all the avenues in which you can explore. All right. So for the practical, I have included also a Xenium data set because I think you need to understand. All right. I mean, if you're doing spatial, and Xenium is an example of a single molecule imaging-based platform, and this is a sequencing-based platform. The scale is different still, even though both of it measures subcellular resolution. So I think to give it some context, I've downloaded, you have the mouse data set for the Xenium, all right, that's already downloaded, all right? And I want you to try both, all right? And just see for yourself, even if you're not doing Xenium, even if you're not doing Merscope or CosMX, it doesn't matter, all right? Seeing is always, they say seeing is believing. So if you see it, you'll appreciate it more. So imaging-based systems. So I don't want to talk about this earlier. When it comes to imaging-based system, what are you really looking at? What are you measuring? Is it true that when you image your spots or your probes, you are imaging an actual molecule? Not really, all right? So this is a paper, and I would actually encourage everyone to read this paper. And there are many papers, and you can even read, I think, Stat Microscope is kind of similar as well. Basically, the whole point, all right, in all of these microscope systems or microscope-based platforms is really to resolve your points from wavelengths, from what do you call it, from photons that are emitted, right? So photons have wavelengths, all right? And this is what we call the point spread function, all right, of a microscope. So they use some sort of a prediction probability distribution tools, all right, to make sure that they can discern, all right, between 2 points that are so close to each other. They might even be in the same pixel, all right, and they can kind of what do you call it, identify which molecules these actually belong to. And this is something that is on 10x website. And when I first started my Xenium experiment, I actually had to sit with an officer from their company, and I had to go through and review how to design my probes because I designed a full 414 custom gene. And I was very naive then, and I kept pushing, no, can I have this? Can I have that? And it was a complete no, right? Because the whole point is that just like how many of you do imaging? Right? How many of you know the pain of optimizing and diluting your antibodies so that it doesn't saturate? Yeah. So the thing is, this is fluorescence emission. It's light. All right. Light can be blinding, and light doesn't just blind our eyes. It also can blind the optics of your microscope. So every single point that you see on a Cosamax, on a Merscope, on a Xenium, just know that it's a prediction. So remember a few lectures ago, I talked about how these images, these sequential images are not open source and only Merfish, only from Merscope is open source. What is the value of making these available to you? Because there are different ways to actually analyze these images and to actually come up and predict these molecules, all right? So that is how. So this is all fully imaging-based system. This is how imaging-based systems work in general. And there's a code word. And I'd actually encourage you to go and read this, all right? Because the code word and Merscope, Cosamax, they all have their own kinds of code word. But the code word actually strengthens, all right, how resolved, all right, or the p-value, you can say, all right, to think of it simply, what's the p-value? What's the certainty that the point that I'm measuring is actually a point, right? So there are two things. One is a point must be a point. And the second thing, a point must be the gene that I think it is. All right, so that's image. So everything is fluorescence emission and how much you can actually resolve the distance between 2 point spread functions. And there are limitations to this. Now, when it comes to sequencing-based systems, right? Sequencing-based systems, whether it's Visium or whether it's stereo seq, or whether it is Divid seq, all right, it is all about points. It's all about spots. So these spots are predetermined. It's an array. Think of microarrays. I don't know, maybe some of you are too young to know microarrays. Anyway, yes. So microarrays was where it all started, right? So microarrays, you have this grid, and then you have all your probes, and then you kind of mush up your tissue, and then you just pour it over, and then it catches, right, all the probes. It's the same concept, right? Except that right now we have improved this whole microarray concept, and we are printing the barcodes directly onto these slides. And so these are spots, like I said. Now, what are the limitations of these spots? So there is some people will argue that what do you call it? This is perfect. There's no space between the spots. So for Visium and for stereo seq, for Divid seq, there's dead space in between. But the thing is, RNA diffusion is a reality. It's not true that it doesn't happen, all right? So how much it happens, it really depends on the tissue. So it depends on which organ it's coming from, how it's processed, how old it is, whether it's fresh or whether it's fixed. It does matter. That is the reality. And of course, all these technologies, I'm not doubting these are good technologies. All right. I'm sure the R&D teams have covered all of this and they've tried to minimize. All right. We're not accusing anyone, but it doesn't mean that it doesn't happen. All right. It does happen. So these are some of the things that you need to consider for sequencing-based system. And the way these spots work, all right. Now, if you want, so the resolution when it comes to spots, so when it comes to sequencing-based systems that rely on spots, is really the distance between two spots. So it's not because the square is 2 micron, it's because the distance between the center of two squares is 2 micron. So for a stereosig, all right, the spot, I think, is 250, but the distance between the spots is 500 nanometers. All right. So that is the closest that two cells, all right, for instance, let's give an example, can be close together for us to resolve it. Again, the principle is very much borrowed from image-based system, because that is how we actually determine what is the resolution, right? And I think these are things that you have to understand, all right, before you talk about how much you want to deconvolute, whether you need to deconvolute. I remember when I was at the neuro and I used to spend hours running my flow cytometry, and there was this person beside me who would always sort astrocytes, all right? The astrocytes are much, much bigger, much bigger, all right? If you're dealing with just astrocytes, all right, a tissue that's full of astrocytes, what is your resolution going to be? How much do you need to bin, right? So these are things that you can actually adjust for, that comes from your knowledge, because you know your projects best. You know projects better than any of the technical officers from any company. So yeah, use that to drive things through. So here, all right, the scale here, this is an example, so I had this then. So the spot size is different, and the center to center measurement is what gives you the resolution, all right? So make sure you go in and you get all these specs from whatever tag that you're using. And I know that I keep using the StereoSig as an example. I do like the StereoSig. I think it is also in many ways comparable. And it's also easy to teach because there's things like spot size, dead space, and all other things. So it's easy to talk about such things. Now, when it comes to scale, all right, this isn't exactly scale, I guess, but I'm going to talk about dead space. All right. We are all, I mean, who likes dead space, right? It means that you're missing something, right? But if you're doing something like profiling, you're not really interested in minute reductionist questions like cell-cell communication or interaction properties. Does it matter? All right. We know that in principle, all right, when it's like random sampling, if you're randomly sampling a tissue area, does it matter if there's dead space? If the tissue area is large enough, you're going to capture enough spots from enough regions, all right? And you're going to be able to profile, all right, the sample features or the region features, unless, all right, the region that you're trying to discover, all right, is really small. All right, then of course, even then, right, it depends. Like I wouldn't recommend maybe a Visium. I'll definitely recommend a Visium HD or a StereoSig, right? But if you're going to do a very holistic profiling, all right, of something, of a tissue, all right, that's large enough, you're not really interested in studying any of the minute reductionist details, then Visium would actually suffice, right? So when you know, all right, the range of your scales and the range, all right, of the tissue that you're investigating, all of this will actually guide you. So yeah, how do we define scale? We can also define scale as the ability to look at single cells. For me, single cells are very important, but that is because I'm looking at geometries. I'm looking at population geometries. So single cell is very important. I need to understand my single cells. Someone who's doing cell-cell communication, single cell is also important. All right. Now, whether do you need a segmentation or not, I'm not really sure, because you can study cell-cell communication using segmentation-free methods. It is still an area of science that I think is still undergoing development, but it is possible. Like I've used BESO, I've used ProSeq. These are all pretty, BESO especially, it's a segmentation-free method. You don't really need to do any segmentation. It gives you tissue profile, it gives you subcellular aggregates of transcripts, and maybe that is all you need, right? Like, so giving an example from protein. So if you look at protein, if you look at frag assays, if you look at proximity ligation assays, all right, you don't need cell boundaries. You don't need to draw cell boundaries. You just need to know if two clusters of molecules, all right, are in the same cell, in the same vicinity, and if they're close together, right? So it's something like that. So in this case, both of these, Stereo, Seek, and Visium HD, they have provided code, all right, where you can do your segmentation, all right, but the segmentation is not accurate, as you can see, all right. So they use the nucleus as the anchor point, and then the cell area is estimated. You can stain your tissues. As Amina has mentioned, most of it is H&E. I've also not seen anyone try out fluorescence and then run it through what do you call it, a Visium HD. It is, okay, so this is something, some people will argue you can do an exact sister section, do your staining, and image register it. We all know that tissues change very, very quickly, even if they are 5 micron thick slices. All right, especially like for myself, I work on GBM. It changes so fast. All right, I wouldn't even bother to even expect that the nuclei is going to overlap. All right, so just know, all right, that all of this is going to be something of an issue and something for you to consider, especially when it comes to spot-based assays. Now, this is an example of single molecular imaging analysis or output. All right, so this is an example from the Xenium, but this is what you'll get even if you use the Cosmax or even if you use the MERSCOPE. All right. So the wonderful thing about these imaging-based systems is that the transcripts really do help. I think they really do make a difference. But again, do you really need it? All right. So in this case, what is my scale? All right. So here, all right, I have more or less a bigger scale. My scale is either the size of my bins, all right, or the size of the smallest unit of measure. In this case, my scale becomes the size of the cell, all right, that I have what do you call it, derived, all right, from any one of these segmentation methods or pipelines. And here, all right, my scale has become the transcript itself, right, or the point itself, I should say point, all right, that point that you see that is supposed to represent a transcript. So which scale is important to you? All right. So these are things that you have to think about. And that's how you see scale. So scale is it is driven by the platform itself, which is the tech spec, right? So you can download the technical spec sheets, you can understand all of this. And then next, it is also driven, all right, by what is it that you want to query? What is it that you want to query? All right. And scale, we already discussed scale when we talked about spatial clustering. That is also scale, but it's just a scale that is screening over a broader region. So when your scale becomes bigger, like bulk RNA sequencing, that is also all right, a scale, right? In this case, your scale is the whole tumor chunk, right? And how you define that scale will determine what is the biological through output or input that you want to measure or you want to give. All right. And what did I want to say? I want to say, oh, and also when it comes to segmentation, right? There are some types. So there are some grids. There are some types of grids where it is easier. That is going to be easier to segment. So I talked about the nature of the tissue. All right. So if you have really large cells, it's actually easier. All right. But if you have really small cells, like immune cells, it's going to be difficult. If you're going to deal with bacteria, it's also going to be difficult, right? So know all these things. All these things will affect. And Stereo 6 suffers from the same resolution problem as Visium HD, but Stereo 6 is a little easier actually to classify or to aggregate it by the cell area because of the fact that the spots are small as well. So think about all these things. And then, yeah, this is just an example where you can simply look at the distributional patterns of your genes as opposed to simply looking at cell features. So there is, so previously in the previous, no, in module 4, Savannah introduced the concept of gene modules. You can identify gene modules, all right, and you can understand how these gene modules are distributed across space. You don't need single cell, and you can just make do with any scale as long as, all right, the bin size that you choose is sufficient enough for you to pick up the variations in the gene module that is of interest to you. All right, so these are things that I think is important when it comes to measuring scale. All right, so this is going to be quick, all right, and no more code, all right, no more like lectures with like details and papers or anything like that. It's just going to be an overview, a review, all right, of the past 2 days, all right, so you can just relax, listen. So yeah, so this is just wrapping up and some things to think about and to just remember. And also because I prepared this slide, um, because there are a lot of interesting questions and these questions are good questions. And like I said, it is always more important for everybody to ask, all right, questions that people don't like asking, all right, about, because that is where you actually draw boundaries, right, in how and what to investigate. So for the garbage in and garbage out module, all right, the whole essence or the whole, even though we don't really have a lab practical for that, the whole essence is really to make you understand. It all starts with that instrument. It all starts with the tissue, right? Handling an instrument is also science, all right? And if you think that it's not, then go and talk to Amina. She will let you know, all right? It is science itself. There's a lot of things that they need to learn, all right? A lot of things can go wrong with the instrument that can give you data that is just spurious, all right? And there's a lot of effort, all right, that goes into all those little region packets, all right, those region packs that 10x sells you or any company sells you, right? So if you refer back to your molecular biology courses, right, you know that ligation, reverse transcription, hybridization, your GC ratios, all of this matter, which means that, all right, what you are capturing, all right, at the transcript level is dependent on many of the caveats or many of the shortcomings that we have learned in molecular biology. So as these enzymes improve and they get better, all right, your platforms will change, all right, and what you can gather out of it, all right, also changes. So try to understand all of this because it's important. So convenience is good, but when we over rely on that, we lose control. So we are all very used, to have all these standard packs, to have someone like Amina doing all the work for us, all right? But the thing is, we cannot forget, all right, that any one of these steps can be optimized. The reason why I say this is because it's also important to read papers out there where they are dismantling all these packages and they are fine tuning, all right, and they're playing around with the regions. So there are labs where they will take the kit and they will repurpose it. All that can be done. All right. If you need to improve, all right, your reactions, it can be done, but it just means that you have to do the job. All right. It doesn't mean that if it's packaged, it's the end all for all our analysis. Now, know your tissue. All right. So Melanie Peralta went through that whole process. So why is it important to know your tissue? If you don't understand your tissue, how are you going to judge it? How are you going to make an informed choice of the platform and of the analysis methods, the process itself? And every organ has a unique chemical composition. So you have one set of barcodes imprinted on a slide, all right? And you have to use it for every single one of your tissue, right? We all know, all those who have done immunohistochemistry and sectioning, we know that tissues vary in how well they even adhere to the slide, all right? The age of the tissue matters, all right? So if your tissue is not going to adhere well, it's not the machine's fault, it's not the technician's fault, but maybe that is a limitation on your tissue and you just have to choose something else, all right? And the content, how your cells are fixed or how your tissue is fixed, is also important. Some people do collagen, some people do FFP, some people have to take frozen because it has to go through a surgical time point, right? So these are all important things to remember. That is why we make you go through that morning full of lectures so that you understand and know all this. You don't have to be experts in it, all right, but you need to know something, right? So think carefully, all right, also about the range of cell types you expect from your spatial versus non-spatial experiments. And I would actually encourage you to talk to your specialists about this, all right? Get to know what they are doing, all right? And very often you will learn a lot more, all right, about your experiments when you talk to them because they have experience that is incomparable, right? And that goes to every single one of you. You all have individually have experiences that we all can learn from. So quality control. So quality control is not just because of poor quality data. Quality control is also about the technical specs of your platform. So I can give one example. The quality of the run was really high for one of my Xenium runs. But during the actual filtering process, all right, I had to completely discard a tissue because it was over dense, all right, on one section, and the points were too close together, and the fret quality suffered, which means that it made me doubt whether I could actually get something that was reliable in that region or not. So these are things that sometimes is just the nature of your tissue, and you just cannot push the system anymore beyond that. All right. And there's another example where the tissue was just too overinflated because of improper fixation. So this is a technical issue on the tissue fixation side, but the run was successful. But it's just that the counts per bin was just too poor for me to extract anything viable. Right. So quality control can be attributed to different types. All right. And then normalization. Now, when we talk about normalization, the first thing you have to ask is, why do we normalize? Everybody says why do you do log normalization? Is it because it's the standard? We need to understand why we do normalization. The whole point of normalization is because we do not want underrepresented genes to be lost, all right, when we do our analysis, and we do not want overrepresented genes to consume our analysis, right? So that is why we do normalization. In many imaging experiments that I've analyzed, I don't go through log normalization. I simply filter and then I do a Z normalization, all right? So you can try different varieties of it. You can try acid transform. Acid transform is very, very computationally intensive. You can do Z normalization, and you can also try Freeman Turkey. All right. And there's no rule saying that you cannot do a simple Z normalization. And Gary is here, and I'm not done with my presentation. Oh no, right? Okay. So so normalization, all right, I would suggest you don't just stick with log 1p, all right? The counts, the sparsity for a Visium HD is very different from single cell. So I think you might do well even with a simple Z normalization after filtering out all the too high and the too low counts, all right? So try it. And then for module 3 and 5, all right, why do we go through the whole process of spatial modeling? And why did Alyona take you through the whole process, all right, of overlaying your clusters over an image, right? So the thing is, during module 3, what I didn't talk about were things like why do we perform PCA, why do we use UMAP, and we didn't talk about too much about what are the dimensions that we choose. In general, the principle is the same, all right? But the question is, do we need PCA, all right? So the function of PCA is, well, there are several reasons to do PCA, right? So one thing is you want to dilute out the noise. So you want to denoise your data set, all right? You want to enhance the actual biological signals. But some people also do PCA because it is also good and it makes your compute process efficient as well. So it's also for resource management as well. There are papers where they have shown that it doesn't really make much of a difference when you don't perform PCA. All right. So think about all these questions when you perform your downstream analysis. And then understanding spatial clustering. So lots of questions, right, along the way. Why do we need spatial clustering? And it's true, we don't need it all the time. If you already have a brain anatomy or your anatomy of your kidney all well outlined out, why do you need to even do spatial clustering? Now, for one thing, spatial clustering is very convenient. All right. If you notice the process that Alyona took you through, it is tedious. You need to draw out the anatomy. All right. It is tedious, but sometimes that is what you need to do because you have a specific region, all right, in your model that you want, all right, to be compact against, especially in differential gene expression. So don't just do spatial clustering because it's there. All right. Also think about what is the benefit of using spatial clustering. All right. Do you want to discover? Do you want to do something? And even if you want to outline all the spatial clusters, which is anatomically relevant, you still have to cross-check it, right, with your Allen mouse brain atlas. And it might just be easier for you to download the mouse brain atlas, use the annotations, and just transfer it over and use that as your spatial clusters. It's all there. So it's how you drive spatial clustering, depending on what questions you want to ask. And then for module 5, that was the process that Alyona took you through, right? So for any kind of analysis where you have to discover something, so both me and Alyona, we are working on like discovery approaches on our tissues, okay? So for us, it is also important that we don't over discover, you know. Sometimes you see a cell type and many papers will say that, oh, that's a rare cell type, but you know, maybe it's just something that we want to see, right? So we don't want to hallucinate like how AI can or ChatGPT can hallucinate sometimes, you know. So that is where you look and review through your H&E. You look and review through your image. The H&E is there. You're using the H&E to pick your ROIs, right? So you pick that ROI for a reason because it was an interesting choice. It was an interesting region to pick. So why not just use it because it is there, right? And so one of the things is, right, many of these, as I mentioned before, many of these paradigms, many of these pipelines, they're actually borrowed from the imaging world. Like every imaging scientist has gone through this. They have stacked their image, they have outlined their ROIs, they have gone through the whole process of what scale how scale has an effect on their analysis. So always remember to this is spatial, all right? So spatial means you have to go back and look at the distribution in your real time, in your real space itself, all right? So balance between the two and talk to imaging scientists, all right? They'll probably have more to tell you and advise you about scale than me or maybe most of us here. All right. And I've learned a lot by talking to microscope specialists and so on and so forth. Now, in module 4, gene inquiry and visualization. So the question is, why do we perform differential analysis? And how much can we deviate from tradition? So when you do differential analysis, why do you want to do? Is it really because you want to look at fold differences? Or do you just want to identify cell types enrichment in a region? There are different ways to do it. You can also look at PC loadings, all right? So you can just look at a PCA plot. You can look at the top and bottom PC loadings, and you can investigate whether there's any sort of gene enrichment in any of these cell types. And someone asked the question if you can subcluster, all right? And you can just look at the subcluster groups and you can do a differential analysis. You can do that. If for most of you who have done single cell experiments, that's what we used to do, right? We used to do clustering, PCA clustering, and then we would divide up the cohort into immune and tumor or normal cells, and then we will subcluster again. So before you do any kind of differential analysis, also bear in mind what are your target groups or what are your target cell types, and then subcluster them and then investigate them. So all these pipelines is a general pipeline, is for you to use and guide, all right, so you can kickstart your project, but also feel free, all right, to manipulate your data set as you see fit, all right? What you subcluster and what you investigate, all right, and the regions of choice, it can always be a hierarchy. So you can always form a hierarchy. It doesn't mean that if you normalize once at the beginning, you cannot subcluster and renormalize and recluster and do another differential analysis. And then for computational scaling, it's really to talk about the 2 micron, 8 micron, 16 micron bins that we find in Visium HD, all right, and to also critically understand what motivates your bin size selection. The 2 microns suffers from noise. It really does. So as much as it can, the system can actually evaluate 2 micron bins, all right, but the power of evaluation does not actually lie in the 2 micron bins. It lies at much higher bin sizes, all right? And there are ways in which you can define these bin sizes. So you can define these bin sizes, all right, by the cell itself. So you can do cell segmentation, but the cell segmentation method is going to vary between the platforms. 2 micron might not be sufficiently small enough for really small cells, which means that your segmentation boundaries and your bin size will suffer. If you want to aggregate your bins, all right, always remember that if you just aggregate them and you want to do single cell, then you will suffer from mixed cell signals. But if you're not interested and you want to do broad domain specifications, all right, then it's fine. But if you want to do very granular single cell subtyping, then you have to, I think it's worth trying segmentation, all right, but also know that it is limited by the nature of your cells that exist and the potential of your smallest unit of investigation. So in the previous workshop, a lot of people asked the cross applicability of all of these methods and concepts. They are all broadly applicable, but there will be differences, right? So all of these are spatial methods. You're looking at spatial relationships, distributional patterns, but there will be differences because there are inherently differences in your chemistry and the type of probes, all right? Which means that your number of counts, the extent or the distribution of your genes, all of this is going to vary. And of course, all of this is going to affect what type of methods. Some methods are better in some platforms. Like for instance, Basol works very well if you have a merge code data set, which is what I have found, but it didn't work for me with the Xenium. It was just over segmenting, or it was under segmenting. It was very often seeing doublets as opposed to singlets, right? So these are things that is not it's not an issue with the package, all right? So when you read all these methods paper where they benchmark and then they criticize what do you call it, package A or package B, it might not be the package, it just might be just that the parameter fine-tuning isn't up to what it should be, or it could just be that that package is just suitable for some technologies and not others. All right. And then, yeah, I don't want to yak too much because I know you guys are tired. And yeah, I'll leave this open for any questions. And then we will have Gary come over and give the final.